Hello, I'm Todd Sacktor, a SUNY Distinguished Professor at Downstate Medical School. And my laboratory has studied how long-term memories are stored in the brain, which has been a fundamental mystery in biology and medicine. And we know very, very little about how memories are stored in the brain. We know the brain contains 86 billion neurons, and each of those neurons is connected to between 1,000 and 10,000 other neurons. And those connections are called synapses. And these synapses can be strengthened or weakened by the activity of the neurons. But a fundamental question is how that strengthening persists over time. And that's key to how memories are stored, because it is thought that a single memory is a population, what we call an ensemble of neurons. We don't know how many, maybe just a few thousand out of the billions, that represent a single memory. And so the fundamental question for memory storage is, how are those synaptic connections that tie together this ensemble of neurons maintained over long periods of time? Now, we actually know a fair amount about the molecules that do that strengthening. And that's because memory is known to go through various stages in time. There's a short-term memory, which lasts just a few minutes, that if it's important enough to the person or non-human animal, gets consolidated into a long-term memory. And that consolidation process lasts a few minutes to a few hours. And we know a lot about the molecules that are involved in that transition from short-term into long-term memory. But what we do not know is, what is the molecular machinery that keeps that memory going from a few hours to a day to a week, and for humans for decades, a lifetime. So that's the fundamental question that our laboratory has been investigating. And the way we started is by making that assumption that what's important is the strengthening of the synaptic connections between neurons and what makes them persistent. And so that became our first question as well. What is the molecular machinery that keeps a form of synaptic plasticity called long-term potentiation, or LTP, going? And there the question is the same as in memory. We knew a lot about the molecules that were involved for the first few minutes of long-term potentiation, or LTP as it's called, but knew nothing about the molecules that are maintaining the LTP for many hours, days, and even in animals for weeks. Now, as I said, we knew a lot about those molecules that are initially important for LTP. Hundreds of molecules turned out to be important. We also knew that new gene expression was an important key feature for the transition from short-term memory into long-term memory. The same thing was true for the potentiation of synapses. The transition from an early phase of LTP into a more persistent phase also required the synthesis of new proteins. But when people looked at the genes that changed their levels of making proteins, it looked like 14% of the genome was changing. Thousands of genes probably changed. So most scientists basically gave up the search for a simple underlying mechanism for how LTP might be maintained, and thus how long-term memories might be stored. But when I came to SUNY Downstate, I was interested in a very particular, rather simple model of how an enzyme could be made persistent. And I thought that might be the key for understanding how a short-term memory might be made persistent into a long-term memory. And that was because enzymes can regulate the strength of synaptic transmission. One of the most important enzymes for regulating the strength of synapses was called PKC. So here's PKC shown on the left. It has a regulatory domain in red, and I'm showing it a catalytic domain, the action part of the enzyme, in green. And normally the regulatory domain inhibits the catalytic domain. And then the enzyme gets activated when second messengers bind to the regulatory domain, causing a change in the shape of the molecule, releasing the autoinhibition from the regulatory domain, allowing access to substrates that then the enzyme could then work on. But it was known from the very first papers of PKC that there was a way to permanently activate the enzyme so that the second messengers, which only last for a few seconds in cells, became unnecessary for the enzyme to be active. There was a way to make a persistently active enzyme. And that way was basically to separate the regulatory from the catalytic domain by another enzyme that split the enzyme in half. And the release of the catalytic portion created this independently active enzyme, which was called PKM. Now, when I started my laboratory at Downstate, there was no evidence for PKM in a cell. It had always been a test tube phenomenon. So my first goal was to simply see whether there was any form of PKC that got turned into a PKM form. So by that time, in the early 90s, when I was starting my laboratory, it was already known that PKC was not coming from a single gene. It was actually coming from 9 different genes. So what I did is I made a way using antibodies to identify all 9 proteins, all 9 forms of PKC. And what we found was there was only one form, the zeta isoform, that had this fragment. We can see here is lower down on this gel of hippocampal tissue. So there was really only one form of PKC that had a PKM form. So we focused on that. And what we found was when we induced long-term potentiation and saw a strengthening of the synaptic connections between neurons, there was also an increase in the amount of the PKM zeta. That was also very persistent. Now it turned out it was a lot more complicated than we thought. It wasn't the simple mechanism of proteolysis, of splitting the PKC zeta into PKM zeta. It took us 10 years to figure it out, but it turns out that the PKM zeta is made by new synthesis from its own gene product. And that's why the persistence of PKM zeta, like the persistence of LTP, required new protein synthesis. So then the next step was to see if we could inhibit PKM zeta and see if the potentiation of the synapses could be reversed, because nothing had ever previously been shown to do that. I mentioned that there were hundreds of proteins that were involved in LTP, but they were only involved in the first few minutes. If you inhibited them Hours later, they did nothing. But we made an inhibitor of PKM-zeta by taking the piece of the regulatory domain that was absent from PKM, synthesizing it, and changing it so that it could now penetrate into the neuron and act at the synapses. And what we found was that when we induced LTP, we could wait hours afterward and then add to the neurons this small drug, which we called zeta inhibitory peptide, or ZIP, and it reversed long-term potentiation without affecting the synaptic strength of synapses that had not been strengthened. So this was a very unique phenomenon. No one had ever seen or had a tool for reversing LTP once it had been established. The next question was, if we have this tool for reversing LTP, can we use it to erase a memory? We then had to figure out a task which was very similar to LTP. We work in a part of the brain called the hippocampus, which is important for the initial formation and storage of memories. For rats and mice, this part of the brain is important in particular for the formation and storage of the knowledge of where the animal is in an environment, where its place is. So we took advantage of that by having a task in which the animal could rapidly learn and then remember where it was in a particular environment. So what the task, which was invented by my colleague Andre Fenton, works like this. If you put a rat or mouse in this rotating arena, it can be brought into a pizza wedge-shaped sector which is not rotating. And in that sector, the animal can receive a very mild shock, just enough for it to want to move out of that region. The thing is that the apparatus is rotating. So the only way that the animal could learn and remember to avoid this non-rotating sector is to pay attention to the cues in the room. And this task takes advantage of the function of the hippocampus. And it forms, and the hippocampus is important for forming and storing this memory. So we knew now where in the brain to inject the inhibitor of PKM-zeta, ZIP. So here are the experiments that we did. We first took a rat and put it into the apparatus. So the rat tends to spend its time on the outside of the apparatus, maybe looking through the clear plastic walls. And this gray zone is where the shock will be. Right now, the shock is off. So to train the animal, we turn the shock on. And we put the animal in for 10 minutes, take it out for 10 minutes, put it back in, and repeat that 8 times. By the end of the 8th training trial, the animal, as you can see, has learned to avoid this shock zone, which in this video we show as red. So we then take the animal out for a day. And then for control animals, we injected into the hippocampus just saline. It's a control. And we put the animal back in with the shock off. Again, we're showing the region where the shock had been in gray. As you can see, the animal still remembers to avoid that shock zone. Then we took another group of animals, trained them, waited a day, and injected ZIP into both hippocampi. Put the animal in with the shock off. As you can see, the animal seemed to have no memory of where the shock had been. The animal's memory appeared to be erased. Well, this was a remarkable result. And so after we published it, a lot of other labs around the world wanted to see if this was true. And it turned out that the results were very consistent among labs all around the world. That it wasn't just this active place avoidance task, it was other types of memory that depended on the hippocampus, memories that were involved with fear in the amygdala, memories of skilled motor learning, something analogous to learning how to ride a bicycle or play tennis, which can be stored in the motor cortex and sensory cortex, habit memory stored in another region of the brain called the striatum. All of these types of memory were erased by this inhibitor of PKM-zeta. And it wasn't just the inhibitor ZIP, it was several other ways of inhibiting PKM-zeta. And even though the ZIP, they all erased memory. And even though the ZIP in the brain lasts only for a couple of hours, it could disrupt memories that were 1 day to 3 months old. Short-term memories, those memories lasting only a few minutes, were not affected at all. No memory retrieval was necessary. In other words, you didn't have to recall the memory in order for it to be erased. And thus multiple memories were erased. And there appeared in most cases to be no spontaneous recovery of the memory. And after the drug was washed out and memory was erased, the animal could nonetheless relearn and remember normally. So it wasn't as if the brain had been damaged. It was like a hard disk that had simply been reformatted so that all of the information was gone, but the hard disk itself was not damaged. We've done further experiments now to visualize the PKM-zeta as memories are formed and maintained in the brain. So for the first time, we can now see where the memories are formed. On the left is a control animal, and on the right is the animal after it has gone through the active place avoidance 1 day later. Again, there has never been a molecule in which you can actually see where the memories are being stored. We can actually look even more detailed level to see which synapses that are containing the PKM-zeta. So here we see in panel C that there are many spines coming off of this dendrite, colored in green, but the PKM-zeta is only in a few of these spines, and that's this yellow next to the arrowhead here. So we can actually then count the number of spines which contain the synapses that are maintaining a single memory. Lastly, it's not just how memories are normally formed that PKM-zeta is important for. It's also important for psychiatric and neurological diseases that seem to involve memory or memory-related processes. Other labs have found that there are increases in PKM-zeta in post-traumatic stress. In the regions of the brain that encode pleasure, opiates and cocaine addiction cause increases in PKM-zeta. Conversely, PKM-zeta levels are decreased in mouse models of depression in other areas of the brain, including the hippocampus that I just had been talking about. In chronic neuropathic pain, the pain pathways get sensitized in a persistent fashion. And people have found that PKM-zeta increases in these pathways, and just like memories can be erased, the pathologically increased pain can also be erased. We've also found that in Alzheimer's disease, the levels of PKM zeta in the hippocampus are decreased, while in other cells, such as the glial cells that are supporting the brain and the neurons, there may be levels of increased PKM zeta. So in conclusion, we now know the very beginning about how memories are stored in the brain. We know that PKM zeta is a persistently active form of protein kinase C that's formed through new synthesis in LTP and memory formation. We know that the PKM zeta maintains the enhanced synaptic strength of the synapses, and that as a consequence can maintain long-term memory in physiologically normal mice. But that there are also abnormal increases or decreases of PKM zeta that have been implicated in memory-related processes in many neurologic and psychiatric disorders. And therefore, PKM zeta is the leading candidate for a component of the molecular mechanism of long-term memory storage. And there is much more further work to be done to understand this process in detail. And that's going on at SUNY Downstate and other laboratories around the world. Thank you very much.